Hi, my name is Cyril.
And I'm Micayla Lerdal.
And we did a project with Dr. Seth Fritz on the gut microbiome. And our project is called Identifying Microbial Species in Feces, the use of rDNA sequencing to characterize microbes. So, first of all, what is a microbiome? Well, the microbiome is basically the collection of all microorganisms like bacteria, fungus, and viruses that are found within a living organism. A fun fact about the microbiome, bacteria in our body outnumber us from 10 to 1, and there are over 100 trillion bacteria that are found in our body.
Which is really interesting because when we think about our bodies, we think of us as being human made of just cells. We don't realize that we are composed of so many bacteria.
So why study the microbiome? Well, the microbiome is extremely important for human health. It's really important for immunity, and especially it's also very important in our digestive tract because there's millions and billions of microbes found in our digestive tract that help us with digestion and the absorption of food nutrients. It's also extremely important in the environment. It plays a huge role in all ecosystems. It's found everywhere in nature, and these different microbes perform different functions. And recently, microorganisms have also been used for biomass fuel.
Because of ever-growing medical research, the importance of studying the microbiome has greatly increased. So the outline for today's presentation, just to give you a heads up on what we'll talk about, our methods will be first, and it's going to discuss how we identified our microbiome. So what steps did we take in order to establish the research? We will then talk about the results. So we'll talk about the fecal DNA extraction, the rDNA amplification. We'll then also talk about the cloning and the sequencing of the DNA in order to identify the bacteria inside the rat feces. After we'll wrap up the presentation with the conclusion with species in the feces.
So for the microbiome analysis, what we first did was we extracted DNA from the fecal samples, and we used a kit called a Zymogen DNA extraction kit. Once the DNA was extracted, we ran it through a polymerase chain reaction, also known as a PCR. What a PCR does, it amplifies a specific region of the DNA, and it uses these things called primers, which latches onto the specific gene and basically makes numerous copies of this one specific gene. So we focused on two genes. We focused on the 16S rDNA and the 18S rDNA. And the R stands for ribosomal DNA. The reason why we chose these two regions is because ribosomes are found in all cells and they perform the same function. They make proteins. Therefore, the DNA won't vary much among the different species. However, so the small variations that are seen can help us distinguish the different species. So the 16S rDNA is found in all bacterial species, and the 18S rDNA is found in all eukaryotic species. So once we did the PCR, we took those amplified gene fragments and did a transformation process. The reason for this is because the DNA that we have came from multiple different bacterial species. So, and what we want to do is we want to isolate certain sequences to get just a handful of bacterial species because we want to identify a couple of them. So what we did was we inserted these gene fragments into a plasmid, and a plasmid is a circular piece of DNA, and that's basically what DNA looks like in bacteria. And we inserted these gene fragments into this plasmid, and then the plasmid was taken into the bacteria. And each bacteria took up one plasmid. So that means this one plasmid, this one bacteria has DNA of one specific bacterial species. And then once we grew the bacteria, we extracted the DNA from specific colonies that grew on the plate, and then we sequenced that DNA, and we ran it through a BLAST, basic local alignment search tool, and we found our bacterium species.
The BLAST tool is a government-run website, so it allows us to go on there and see the research of other people inside the scientific field, and it compares our sequences, so like the CTA, TAG, and it will use that order and find other sequences similar or the exact same, so we can identify what microbiome species we have, what type of bacteria we have, and it's very helpful. Our results. Can we successfully extract DNA? Our answer to this question is yes. We used a nanodrop after we had extracted the DNA to measure the concentration and the amount of DNA within the sample. Our goal was to actually have over 100 nanograms per microliter, and we pretty much beat that, as you can see. Our first sample actually had 165.1 nanograms per microliter, which is pretty substantial. Our purity, or the A260-280, was not applicable because we weren't able to get the result for that one, but we're still pretty proud of that sample. Sample 2, we had 176.7 nanograms per microliter, and it was the A260 to A280 was 1.80, which is actually pretty good. You always want your A260 to 80 to be above 1.8. That's the level of purity. It makes sure it's making sure that it's DNA, that you don't have goose in there. Our sample 3 had a concentration of 199.1 nanograms per microliter, which is almost double of our goal. So that one's pretty awesome. And our A260-280 for that sample was 1.86. So very interesting numbers up here.
So after extracting the DNA, like I said, we ran it through a PCR. Well, in order to see the PCR results, we had to run it through a gel electrophoresis. And what a gel electrophoresis does, well, it's a method that measures the length of DNA fragments. So in PCR, like when I mentioned those primers, the 3 primers that we used were the 515 forward 806 reverse primer, the 533 forward and 1492 reverse, and the NS1 and NS2. The 515-806 and the 533-1492 both targeted the 16S region, and the NS1 and NS2 primers targeted the 18S DNA. So what a gel does, DNA is negatively charged, and when DNA is inserted into this gel, what happens is the DNA will run through, basically travel towards the positively charged end of the gel. And the further that the DNA travels, the shorter the smaller the length of the DNA is. So each band represents a DNA gene fragment. And so the first band that you see, right there, this is just an inverted picture of that. So the first band that you see is approximately 300 base pairs long. And then the second fragment that you see is approximately 900 base pairs long. And the third fragment that you see is really faint, but that's a fragment. That's approximately 550 base pairs long. And that's good because those were our expected DNA fragments. And that tells us that the PCR was successful.
And the way we get the base pair length is if you look right here, you'll see a ladder. We call this a ladder. And going from here all the way up goes from 100 to 800, and then it'll go from 800 to 1,000, then 1,500, then 2,000, and then 2,500. So kind of restating what she said, the higher up on the gel, so if it's up here, it'll be closer to 2,500. If it's down here, it'll be a lower number, so it'll be smaller.
So after doing the PCR, we transformed the bacteria. And so on this plate, so on this plate, that was a negative control. So the bacteria in this plate didn't contain any plasmids that had inserts because we never added any inserts. And so on the plasmid, there's this gene called the ampicillin-resistant gene. So when the inserts bind to this plasmid, this ampicillin-resistant gene will be activated. Because the inserts were added, the ampicillin-resistant gene can't be activated. So in the presence of ampicillin, these bacteria shouldn't grow. And the plate shows that it didn't grow, so that's good. And then the second plate, this is a positive control. The kit that we used gave us a positive control just to make sure that the ligation and the transformation worked. And it's really hard to see the colonies just because there's a white background, but there are colonies there, and that's good because that tells us that the transformation was successful. So this plate contains the plasmid with the 533 1492 insert. This plate contains samples with bacteria that contain plasmids with the 515 806 inserts, and this plate right here contained the inserts that had the NS1 and NS2 samples. And you can see the colonies better here. Little dots.
So the results, after we had sent in our sequencing to a laboratory called GeneWiz, they sent us this information back. And they said, hey, so we found 2 different kinds of bacteria. Pretty awesome. And then we also found 1 fungal species. So the 1 bacteria, or the 2 bacteria that we found, were the Neisseria vestiformis. This was found in the membranes of an animal's mucus, or in the mucus membranes. And we're kind of interested, how did that get there? Because we're dealing with rat feces, which comes from the intestine. Why is there upper respiratory tract bacteria in there? We also found the Neisseria flava, found in the upper respiratory tract. So 2 different things from the upper respiratory. What we're kind of wondering is maybe did the rat breathe on the feces? Did it get contamination? What is this doing in here? And kind of something really cool to think about. I love the shape of these cells. They're pretty cool. And of course they stain them so you can see them better. But the Bacilliformis seems larger and like tighter together. These are kind of spread out a little bit, but they're more in colonies, more like what we saw in the Petri dishes.
So the fungal species that we found was Mucorrhythmosis, and that is primarily found in soil. And it's also found in vegetables, plant remains, and at times nuts. And the fact that we found it in the rat feces could probably tell us something about their diet or their environment. And yeah. So for conclusions, we were able to extract DNA successfully. We reached the concentrations that we wanted. And we amplified the DNA, the specific gene fragments that we wanted. And the bacterial cloning was successful. We grew colonies on each plate except the negative control, which was expected. And we were able to sequence the DNA and run it through a blast and to identify the species. These are acknowledgements. We'd like to thank the entire FSI staff, Lori Ball, Marie Woods, Amy Dunn, and Sean Hoskins. And I would like to thank my sponsors, the Cyril family, my family, for sponsoring me to do this program. And I'd personally like to thank the ADOP Source Foundation
for sponsoring my research and my ability to come here. That was pretty awesome.
I'd also like to thank Excel Energy for sponsoring my research specifically. And I'd also like to thank Dr. Sepspritz and Ms. Diana Gerard for mentoring us and helping us with this project and basically making this learning experience absolutely amazing.
And thank you to our families and our best friends.